Blood vessels are a set of tubes that transport blood around the body. There are three types of blood vessels. Those are arteries, veins, and capillaries. In the previous video, we talked about a heart. Heart is the pump that pushes blood through the circulatory system, through all the blood vessels. So let's look at the blood vessels in a little bit more detail. As far as anatomy of blood vessels, which one goes where, this is more domain of the lab. We already looked at that, and you have the lab study guide and the lecture study guide that tells you which vessels actually you need to know. There are lots of them. You need to know a subset of the most important ones, most important arteries and veins, and you need to be able to recognize models and to know uh, where they are, where they go, where do they take blood from, where do they take blood to. One way to divide, classify blood vessels are they part of the systemic circulation or the pulmonary circulation? Pulmonary circulation are arteries and veins that transport blood through the lungs. They start in the heart with the right ventricle pushing blood into the pulmonary trunk, which divides into two pulmonary arteries, left and right, which go to the left and right lung, divide into smaller and smaller arteries, arterioles, capillaries. Those capillaries are hugging the walls of alveoli, where the gas exchange happens. That blood gets more oxygen rich. Then those capillaries gradually fuse into tiny little venules, and those venules into larger and larger veins, and finally ending with four pulmonary veins entering the heart on the left side into the left atrium. That's pulmonary circulation. This is the place where arteries carry deoxygenated blood and veins carry the oxygenated blood. Everything else is systemic circulation. Systemic circulation takes oxygenated blood out of the heart via arteries and brings deoxygenated blood out of from the tissues through the veins to the heart. So systemic circulation is much bigger, and it needs to push a large amount of blood through quite a lot of resistance going through various parts of the body, which is why the left ventricle is much more powerful, has a much thicker muscular wall than the left ventricle. Left ventricle has much thinner wall because although it pumps exactly the same amount of blood, the same volume of blood per each stroke, it needs to produce much lower blood pressure so to avoid bleeding into your lungs. Um, the arteries and veins, as you've seen in the lab on the slide, can be differentiated by the structure of their walls. Uh, the arteries generally have thicker walls uh, and smaller diameter. So the first arteries that come out of the heart, the pulmonary and the aorta, are very, very big arteries. They are also what are called elastic arteries. Elastic arteries means that they have much more elastic fibers than muscle fibers. And that is very, very important. They are taking in a pulse of a large quantity of blood coming right into them, and those elastic walls are going to respond by stretching, and then elasticity means getting back to the original size, and then getting back that elastic recoil is going to help push the blood further. Without it, or if those arteries are You have atherosclerosis, so those walls get really stiff and not elastic anymore. Then the heart would have to compensate by becoming even bigger and even stronger in order to push all the blood through the organism. Uh, as those arteries divide into smaller and smaller arteries, they are gradually losing the proportion of the elastic fibers and thus relatively increasing the number of muscle fibers. So when you go down to medium size and small size arteries, they are muscular arteries, they're not elastic anymore. So when you look at the picture of all this in your book, there are all these tunicas, and anatomy and histology that was all done a couple of centuries ago, and people were specializing. They were heart lovers who called uh, everything something you know with a carry on, something with a heart. So you have. Myocardium, and epicardium, and endocardium. And it's the same thing as tunica adventitia, tunica media, and tunica intima. Tunica intima is really endothelium. Endothelium and endocardium are the same type of tissue and they're continuous with each other. Tunica media, if it has a letter M, then it's probably muscles. Tunica media is the muscular layer. It is the same, continuous with the muscular layer of the heart, the myocardium. Tunica adventitia, that's going to be something that is going to be on the outside, a loose connective tissue on the outside. Uh, when you think about tunica intima, it's always going to be something on the inside. The most intimate circle of friends is your inner circle of friends, right? So that's how I remember that intima is always the thing that is on the inside. So as you go further and further away from the heart, those arteries branch into smaller and smaller arteries, and in the end, the smallest arteries start branching into arterioles. By the time you get to arterioles, there is not much left of all these layers. There's endothelium. There's a very thin layer of smooth muscles around it. The tunica media is already very thin, and there's a little bit of loose connective tissue around it, the tunica adventitia. So that's the last blood vessel that actually has all three layers, but those three layers are thin, and these vessels are very, very small. Once they start dividing, they divide into capillaries. Capillaries don't have everything. Capillaries have only essentially the endothelium. And what is endothelium? That is simple squamous epithelium, right? Flat, thin cells that they may have some holes between them or not. Uh, and then those capillaries which are thin, and so all sorts of things can go in and out of them. Water, glucose, oxygen, carbon dioxide can go in and out of the capillaries, and at the end of the capillaries, they start fusing together with each other, forming tiny little venules. Again, those are just like arterioles. They're tiny, they're thin. There is really only tunica intima there. There's not muscle or anything like that around them. And then they start fusing into larger and larger veins. Small and medium veins have all three layers. Tunica media is thin. There's not much muscle there. And the largest veins are the ones going on back into the heart, like vena cava. They have well defined all of the layers, all three tunicas with all of their layers. Uh, so their walls are much thinner. They don't have that much. They're not as solid, which is why when you looked under the microscope and looked at the cross section of an artery and vein, the vein was essentially completely squished, while the artery was still pretty open. So you could see that difference there. Okay, so. Uh, when I described all of this, this is the kind of the normal situation in most parts of the body. So you have the arteries that are branching into capillary beds, a lot of whole networks of capillaries, which on the other end are fusing and forming veins. A capillary bed is something that's usually found between an artery and a vein. Uh, it's not always that way. Uh, there are exceptions to this. One exception to this is what is called portal circulation, and there are a couple of places in the body where we have portal circulation, and this is when you have arteries going and splitting into a large capillary bed. But then there's another big vessel connecting it to the next capillary bed before we go to the veins. So there's a vessel, capillary bed, vessel, capillary bed, vessel. So there's a vessel between the two capillary beds. That vessel can be an artery or can be a vein, depending on histology, right? You're going to look under the microscope to see what it is. We have already mentioned one of those when we're talking about the nervous and endocrine systems, and that is hypothalamo-hypophyseal portal system. Remember, there was a capillary bed in the hypothalamus that was collecting all those factors that are either stimulatory or inhibitory, and then going just through a single vein, a single artery, into the anterior pituitary or adenohypophyses, and spreading into the second uh, capillary bed, which is then releasing those factors to various cells there and telling them either to release or to stop releasing hormones like growth hormone and prolactin and ACTH and LH, FSH, TSH. So that's one example. Another example we're going to talk about when we go to the urinary system, and that is in the kidney. Kidney has billions of neurons and billions of nephrons. Every nephron has its own little portal system. You have an efferent arteriole that goes into the Bowman's capsule, into the beginning of the nephron, and branching into a capillary bed. Then there's an afferent arteriole that comes out of that uh, capillary bed and goes outside of the nephron and then separates into another capillary bed, which is then following the rest of the nephron. So that's another case where there's an artery between the two capillary beds. Finally, another one is the hepatic portal circulation. You have an enormous capillary bed, which is pretty much engulfing the entire small intestine, which is taking all the molecules taken out of the food by the intestinal cells. And it's taking all of that via hepatic portal vein to the liver, where it spreads into another capillary bed inside of the liver, where all of those nutrients, all of those molecules are then processed by hepatocytes, by the liver cells, metabolized if they're toxins, uh, uh, all sorts of small parts are used to be bigger molecules. Bigger molecules are broken down to smaller parts, whatever liver needs to do with all of those molecules that come from food. And then that capillary bed goes into the hepatic vein and back into the inferior vena cava and back to the heart. So that's another example of a portal circulation. So if there's a single vessel that is connecting two capillary beds, that's portal circulation. We have something else. There's an opposite kind of thing, is that you have an artery, and then you have going a capillary bed, and then you have a vein on the other side of the capillary vein. Well, those capillaries are not all going to be open all the time. They are so-called precapillary sphincters that are tiny little smooth muscles that are closing and opening. And so when they're open, the blood goes from the artery through the capillaries to the vein. When they're closed, the blood doesn't go through there, and it might just go elsewhere through other capillaries, or it can go through what is called a vascular shunt or a thoroughfare. There are places in the body where there are single vessels that are connecting a single vessel to a single vessel, and there's no capillary bed involved. There are no precapillary sphincters. There's nothing there. It's just vessel and another vessel connecting it to the next vessel. And we mentioned those already. Those are called anastomosis. We mentioned them when we were talking about the coronary circulation. Remember that the heart is a very energy expensive organ, and it requires oxygen and glucose and all the nutrients in all parts of the heart muscle all the time, which is why the coronary circulation has a lot of anastomosis. What does that mean? There's a lot of little arteries that are connecting to arteries. So the blood can go through this artery, or it can go to the side and go through this artery. Either way, the nutrients are going to get to the same part of the heart, which means if one of those arteries gets clogged, there is still a way to go around it and provide nutrients to every part of the heart. A similar things happens in the brain. That's another very, very energetically expensive organ that requires a lot of oxygen, a lot of glucose, a lot of nutrients. And how do you do it there? There are arteries called internal carotids. Internal carotid arteries are branches on the inside, so medially, of external carotids, or common carotids are splitting into internal and external carotids. The internal carotid arteries are going to go to the base of the brain, and they're going to start branching, and those branches are going to be connected to each other with anastomosis. So you'll have two branches. They're going to be connected by anastomosis. They're going to be connected to these two branches. There's anastomosis. There's about 5, 6 of those anastomosis there, and that whole complex of those anastomosis is called the circle Willis. If you look up Willis on Wikipedia, you're going to see that he discovered this a long time ago. This is not something discovered 20 years ago. This was more like a couple of centuries ago. You would also learn that what we now know as diabetes mellitus used to be called the Willis disease, a disease of Willis, because he was the same guy was the first person who described diabetes, all of its symptoms. Uh, so learn the circle of Willis. That Circle Willis allows for redundancy. That allows for one of those little arteries to be blocked or not functioning for various reasons, and yet the blood will go around it. Is going to use those anastomosis to get to every part of the brain, so you don't get a stroke. And bypasses all the places that's not working by having that redundancy, having the anastomosis going around. Speaking of all these precapillary sphincters that are opening and closing, they're constantly opening and closing, opening and closing. So there's always blood going through some tissues, but never through all of the tissues, right? So a little blood goes through this muscle, this part of this muscle, but not through the next part. And then this one closes, this one opens. So over time, every part of the body gets sufficient blood, but you don't have it all. Why not have it all? If you look at the capillaries, they're tiny. There are so many of them that their total sum volume is greater than the sum volume of the heart arteries and veins. So, what would happen if all precapillary sphincters would open all at the same time? The heart would beat, push the blood through the aorta, through the arteries, smaller and smaller arteries, arterioles into the capillaries, and all of the blood would end up in the capillaries. There's no pressure there. I mean, it's like 30 or something like that uh, millimeters of mercury getting into the capillaries, and then it would just like die there. the whole blood would be in the capillaries and no real force to push them through into the veins. The blood would never come out into the veins, never come back into the heart, and the circulation would just stop, which means you would immediately faint, and after a few minutes you would be dead. Which is why precapillary sphincters have to mostly be closed and open for a couple, seconds and minutes. Okay, let's let some blood in here. Now stop. Now let some blood in there, and then stop. So it's constantly shunting blood here and there, so every part of the body gets some, but never all at the same time. So once we get that blood through the capillaries, they have to go back by veins. Now the blood pressure there is going to be very, very low, and it's going to be very, very slow. About 2/3, 64 to 67% of all the blood volume at any given time inside of the veins, and not in the arteries or capillaries or the heart. So mostly in the veins because it's going so slow. And because it's going so slow, there is always a possibility, because there's such low pressure, that the blood would go backwards, let's say due to gravity. So a lot of our large veins, especially in the lower extremities, have valves. So if the blood tries going back down, the valves are going to stop him, and then maybe the next pulse of blood is going to push it a little bit more. The slowness of the passage of blood through the veins also often requires, in some places, for us to have more veins than arteries. So you might remember that a lot of arteries and veins go in pairs, right? You have brachial artery and brachial vein right next to each other. You have femoral artery and femoral vein right next to each other. Uh, you have carotid artery and jugular vein right next to each other. Those are pairs. And in another video, I talked about it, how developmentally they follow the same growth factors and grow out together. But remember, 2/3 or 64 or something like that percent of all the blood is in the veins at any given time. If it was just one artery, one vein, you wouldn't have the volume. All the veins have a little bit bigger diameter, a little bit more volume. You don't have enough volume, and some parts of the body would not be drained of blood very well. So we have, in some places, some extra veins. One that you really need to learn about is the great saphenous vein. Great saphenous vein is collecting the blood from the superficial and medial parts of the thigh. Those are kinds of things that are necessary because the blood pressure in veins is so low, so the blood flow is so slow. So you need some extra veins in some places, especially bigger places. The thigh is a big place. Uh, Another thing that can increase the flow, not just the valves holding blood, preventing it from going back down, is what is called a skeletal muscle pump. Skeletal muscle pump is a phenomenon that the contraction of skeletal muscles, let's say in the limbs, especially in the lower limbs, you're walking or running or jumping or riding a horse or anything, doing something with your leg muscles is going to actually, these muscles are going to squeeze the veins. So the skeletal muscle pump is going to pump blood up the veins and towards the heart. That's another way to slightly increase the blood pressure in the veins so the blood can get back into the heart. Um, all the arteries in the systemic circulation are branching off from the aorta, uh, directly or indirectly. So we looked at this in the lab, and you have learned, and you have to learn, what are the first three major arteries. We usually ignore the left and right coronary artery. Those are tiny little arteries that are coming out right at the beginning of the aortic arch. But then you have the aortic arch with three big, huge arteries coming out of it. And those are the brachiocephalic, and then the left common carotid, and then the left subclavian artery. And then the brachiocephalic almost immediately divides into the right subclavian artery and a right common carotid artery. So you end up having two common carotids, one on the left, one on the right, that are going to go up into the head, and you have left and right subclavian arteries that are going to take blood into the left and the right arm mostly. Uh, so I have told you before and in the lab that this is how you're going to see it drawn on every picture in every lab manual, every anatomical atlas, every textbook. I also told you that this is because this is the norm, not normal, the most common situation, the most common configuration in the largest number of people, 70 something percent of humans have that configuration. What about the other 30? The other 30% have other configurations, and almost all of those are completely normal and healthy. You can win marathons with them. You can have one, one brachiocephalic that then branches into four. You can have two, three, four, and those can be different ones, five, six. You can have six arteries coming out of the aortic arch, and it's all good. It's all normal. There is a natural variation, and you can look at all around the body and going to see natural variation in arteries and veins and nerves. They're not different. They're different between different individuals. Uh, you can have two instead of one renal artery, the left or the right or both kidneys. You can have one or two renal veins coming out of the either left or right or both kidneys. That's normal, and it doesn't have any effect on the functioning of the kidney. There is a relatively small but growing number of people who have something called a median artery. Median artery is going in kind of in the middle between the radial and the ulnar artery. I probably don't have it, but some people do, and it's completely normal. It is just not something that's drawn in atlases because it's not usual. It's not the most common situation, but there are many configurations that are not considered pathological. They are normal, and it's a normal part of human variation. So going to all these kind of arteries and veins of various part of the body, this is the part of the lab. My lecture videos are mostly about physiology, while lab is mostly about anatomy. So we're going to now move on to some physiological aspects of all of this. Uh, and so let's talk about the control of blood flow. We already talked about heart, right? So anything that affects the heart rate or the cardiac output is going to affect the flow of blood through the blood vessels, right? So baroreceptors, chemoreceptors, and stretch receptors. Stretch receptors in the tendons and muscles. So those are mechanoreceptors. They're going to tell various parts of the various centers in the brain, hey, we are exercising, we're working hard, we need an increase in heart rate because we need greater cardiac output, greater blood pressure, let's go. So that's going to be an effect on the heart. Those are also going to have some effect on the blood vessels themselves. Now remember how if you take SA node out of the heart and put it in a dish, it's going to beat 80, 100 beats per minute, while the heart itself is beating 60, 70. Uh, so why is it slower? Because the cardiac center in medulla oblongata is constantly sending parasympathetic inputs to the heart to slow it down. Then when you release that, the heart speeds up. Then you add sympathetic, speeds up even more. The peripheral circulation, the blood vessels, are opposite. They are constantly stimulated from their center in the medulla oblongata to via sympathetic nerves to have a particular tone, to have um, a tone of their muscles that is vasoconstricting them to some extent. Adding more sympathetic is going to constrict them even more, greater resistance, higher blood pressure. Or parasympathetic, or releasing some of that sympathetic, is going to vasodilate them. A lot of control is going to be local. Opening and closing of those precapillary sphincters is not going to be told by the brain. It's going to be told by those tissues, like, hey, I'm gasping for oxygen, open up. And they open up. Uh, so there's going to be local chemicals, local signaling molecules that are going to keep opening and closing the precapillary sphincters. So those are the metabolic needs of tissues that are directing the precapillary sphincters and the arterioles to open and close. Uh, of course, the nervous mechanism, stress. We talked about stress in general. Uh, Experience of cold or hot is going to vasoconstrict or vasodilate capillaries in the skin, uh, right? If it's increased body temperature, they're going to dilate blood vessels in the skin so it could radiate more energy, heat energy into the environment, and vice versa. If the body temperature lowers, then those are going to be constricted. You get pale because you're trying to conserve the energy. You don't want to radiate heat into the environment. Uh, there are also hormones, including, for instance, adrenaline or epinephrine. Part of the stress response is going to have the same effect. Um, so there are different ways to nervous and hormonal controls of the blood flow through the various parts of the circulatory system. Uh, so baroreceptors are receptors that are measuring blood pressure. You can find them in what is called carotid bodies, which is the very beginning of the internal carotid arteries as they are branching from the common carotid. But there are baroreceptors in the aortic arch itself, in several other places, and and even in the kidney. They're going to sense, for instance, that the blood pressure is low, it needs to be increased. How do you do it? You tell the heart to speed up and strengthen to increase the cardiac output, but you also tell the peripheral blood vessels to vasoconstrict, especially arterioles, in order to increase the blood pressure. Uh, Chemoreceptors are going to sense um, various chemicals. Uh, how much is there oxygen in the blood? How much is there carbon dioxide in the blood? It's going to monitor pH. How? By measuring um, hydrogen ions or even specific acids like lactic acid. How much is there in the blood? And if there's not enough oxygen, too much carbon dioxide, and pH is dropping low to acidic, that is going to be a trigger for the heart to speed up, but also to increase flow through the periphery. Uh, and then we're going to talk about renin-angiotensin system when we talk about the kidney. Those are all parts of hormonal effects on the circulation and the blood pressure and blood flow through the circulation. What is important to realize is that the diameter of vessels is going to be an important factor here. So when the blood first comes out of the heart, it goes through very large vessels, like the aorta, and then those arteries are going to split. So they're going to branch into two or three at a time, to smaller and smaller arteries. Every time there's a branching event, the two the sum of the two new diameters is actually larger than the previous diameter. So you actually having more and more space. The blood pressure is going to get lower as you go. 120 systolic coming out of the heart. By the time you get to the capillaries, it's down to 30 millimeters mercury. By the time the vena cava brings it back to the heart, it's essentially zero. And narrower the diameter, greater the resistance. And so when you go to the arterioles, this is where there's the most resistance because they're tiny. They have very, very tiny diameter, so there's a greater resistance to the blood. There's also lots of arterioles. There's one aorta, there's gazillions of arterioles. So together, they have a much greater effect than what aorta can do as far as blood pressure. So any kind of a signal that is going to tell all the arterioles in the body to vasoconstrict is going to have a huge effect on blood pressure. They all constrict. There's going to be a big change in blood pressure. If they all dilate, there's going to be a big change in blood pressure down. So that is probably the most important aspect of the arterioles. They are the key kind of level of the cardiovascular system where blood pressure is controlled, which is why they're responsive to different chemicals and different nervous impulses, both local and coming from the brain, coming from the other organs, so they can respond both locally and globally around the whole body to any kind of changes in blood pressure. And I'm going to stop here. Your textbook is excellent, so read the rest there. And anatomy is mostly for the lab practical.